Okay, since photosynthesis isn't on this coming exam, those of you that like to do the crossword puzzles for optional points, we'll just flip that over to the third exam. So I think that there's only, there's 2 for this exam? Okay. So any questions before we change gears?
I always have a question, but I forget it when I come to class.
Okay. Oh, on the study guides, the first one for the energy and enzymes, there were a couple of terms that I did not use this semester. Someone asked me about those Vmax and Km when you're talking about enzyme kinetics. So if there are questions related to those 2 things, you're not responsible for that. Okay? So, what's the significance of this slide? There's a lot of different ways you can approach that, but why do I lead off with this? Come on, somebody.
Because it's light, like light makes a rainbow, and photosynthesis uses light.
Okay, yeah, this is illustrating, this is my kind of lead into photosynthesis. Why is photosynthesis so important?
It makes oxygen and glucose.
It makes oxygen and chemical energy, and the source of energy that's being used is radiant sunlight energy. Okay? What do we call the organisms that can do this process? Those are autotrophic organisms. If it weren't for autotrophic organisms, we wouldn't be alive. Okay? There's actually a kind of a common generality that we make, especially in introductory classes, that goes something like this. If the sun stopped shining, what would happen to all life on Earth?
Die.
It would stop, right? It'd die. Everything would die. Because the plants wouldn't have energy for photosynthesis, therefore they couldn't produce their glucose to then use in cellular respiration to make ATP. And we, being heterotrophs, could feed on the autotrophic organisms or other organisms that eat the autotrophs. Okay? So we talk about this idea of the autotrophs being the producers, and then all the consumers depend upon the producers. Next week, I'll introduce a little wrinkle. It's not something that I'm going to spend a lot of time on, but that isn't entirely true. There are actually a few organisms that are bacteria that don't rely on sunlight at all, either directly or indirectly. And if the sun did stop shining, these unique bacteria could actually survive. So I'll mention those a little bit next week, but not quite yet. So here's some example of organisms that are photosynthetic. And we find photosynthetic organisms all throughout the plant kingdom. We've got some photosynthetic ferns. We've got some photosynthetic algae. Why are these algae a different color than the ferns? I mean, aren't plants green? Why are plants green? Yeah, they have chloroplasts, and in the chloroplasts they have chlorophyll. And what does the chlorophyll do? Why is it green?
Doesn't it absorb like an optimal amount of light energy?
Okay, the color of something, a pigment, is the result of which wavelengths of light it absorbs and which it reflects. Okay? So my blue shirt, the pigments in this blue coloring absorb all of the wavelengths of light except what wavelength? Blue. And that is reflected And then that reflected light goes back to your eyes, stimulates photoreceptors in your eyes. That sends off a nervous signal via the optic nerve to your brain and stimulates a part of the brain that says that's blue. Okay? So these green plants right here have the pigment chlorophyll, which absorbs light energy except the color green, which it reflects. And then we see that color green as the reflected light that enters our eye. So why is this a plant and why is this photosynthetic? Does it have chlorophyll? It does, but it's not the same kind of chlorophyll you typically find in most plants. There are different kinds of pigments in plants, different kinds of chlorophylls, and some other pigments that we call accessory pigments. And you see those in the fall. Okay? In the fall, when plants start to shut down, they start to degrade their photosynthetic pigments in preparation for dropping their leaves. And the first pigment that's degraded and broken down is the chlorophyll. And that leaves behind some other pigments that you're now able to see for the first time. The yellows, the reds, the oranges. Those are called accessory pigments, and they help out in the process of photosynthesis. So we'll take a look at those a little bit too. So here's a little single-celled protozoan, kind of like one of the ones you looked at in lab earlier on. Here is Anabina. You looked at this one. This is a single-celled chain of prokaryotic cells. So you can have photosynthesis both in the eukaryotic world and the prokaryotic world. And then here are some little just single-cell photosynthetic bacteria. So this process of photosynthesis is present throughout the whole gamut of life. Prokaryotes, eukaryotes, single-celled, multicellular organisms. Quick review. Reaction with a positive delta G is you don't need two hands to answer this. Not all of these are correct, but more than one of them is correct. Is a reaction with a positive delta G exergonic? That's endergonic, right? That's uphill. Can it occur if an enzyme changes the delta G to a negative value? Can enzymes do that? Enzymes cannot change the delta G's of reactions. They can't change whether it's spontaneous or not. What can they change? They can lower the activation energy and therefore increase the rate of the reaction, the speed of the reaction, how fast it occurs. Is it spontaneous? Positive delta G? No. Does it proceed from a state of lower free energy to a state of higher free energy? Yeah, it's uphill. Sorry, there's only one correct. That's the only correct one. So it's not exergonic. It can't change to delta G. It doesn't go from low free energy. Whoops. Yeah, it does. Does this? So if a reaction is slowed down by an inhibitor and speeds up as more substrate is added, what kind of inhibition is it? It's competitive inhibition because the substrate and the inhibitor are both trying to compete with the active site. If it's non-competitive inhibition, then increasing substrate is not going to have an effect. Where does substrate level phosphorylation occur? During glycolysis? Yes, it does. During the citric acid cycle? Yes, it does. Electron transport? No. When you see oxidative phosphorylation, what should you be thinking of? What process? Electron transport chain, because remember, oxygen is where the electron transport chain, is where the oxygen is required. That is the only step in cellular respiration that requires O2 oxygen molecules, is the electron transport chain. Okay? So to make ATP using the electron transport chain, that hydrogen ion gradient, do you have to have an ATPase channel protein? Yep, you've got to have some way for these hydrogen ions to accumulate in this intermembrane space to move from their high energy to their low energy, or high concentration, low concentration, into the matrix. You need that channel protein. Do you need a membrane that is impermeable to H+ ions? Yeah, you do. Okay? Because what if that membrane were freely permeable? Then the hydrogen ion gradient couldn't build up. It would be like having a leaky dam. Okay? Water levels would equalize. The hydrogen ion concentration would equalize on this side of the membrane and this side of the membrane. Do you need reduced coenzymes? Yeah, that's the source of the hydrogens that feed off into that electron transport chain. Do you need oxygen? Absolutely. And if you don't have oxygen, the whole electron transport chain backs up. Under what conditions can pyruvate be converted into acetyl-CoA? In the presence of oxygen, okay? Pyruvate then goes into the mitochondria and basically begins the citric acid cycle. Under what conditions can pyruvate be converted into lactic acid? Lack of oxygen, okay? So it's fermenting it, okay? Those are both catabolic processes, okay? Breaking glucose down into smaller units. Under what conditions can pyruvate be converted into fat? Is that catabolic? That's an anabolic process, okay? Pyruvate can be converted into glycerol, which is one of the components of fatty acids. Acetyl-CoA can be converted into fatty acids. That's the other component. When might a cell do that? When might it make fat? When might it favor the anabolic processes? When you're not using up as much ATP, okay? So the cell kind of runs this fine balance between anabolic and catabolic processes, and the Krebs, the citric acid cycle and glycolysis are at the heart of both of those processes. So sometimes conditions favor catabolism, breakdown to get the maximum amount of ATP. Sometimes they favor anabolism, siphoning those compounds off and making other molecules like fats. So there's this balance that's constantly adjusting depending upon what the cell's doing and what its needs are. Okay, yes. Okay, so we're talking about photosynthesis. Basically, for most organisms, including ourselves, the life-sustaining process. Okay? The energy of the sun is converted into chemical bond energy, including glucose molecules, which we can then use as a food source in cellular respiration. Here is the overall reaction of cellular respiration. These are the reactants, and these are the products. Okay? This particular reaction, like cellular respiration, is an oxidation reduction reaction. Sometimes it's just referred to as a redox reaction. Okay. So what substrate in cellular in photosynthesis, is being oxidized? What's losing hydrogen?
Water.
Water is being oxidized, okay? And in the process, oxygen is being produced. Where do those hydrogens go? They're used to reduce carbon dioxide into glucose. And some of those hydrogens go to form new water molecules. So this is kind of an unusual reaction in that water is both a reactant and a product of photosynthesis. But these are different water molecules than these are. We'll see why that's the case in just a minute. This particular reaction has a positive delta G. So the free energy of these is less than the free energy of these. Does water really have any calories? Because that's one of the ways we can represent free energy. No. How about CO2? No. How about oxygen? No. Water? No. But glucose has calories. It has chemical bond energy. So we start down here, and we end up here. Embryonic process. So what's the energy source? What drives this? Sunlight. Photons. Here's a summary of what happens in photosynthesis, kind of showing things in little black boxes. And what I'd like to do is talk about this first to give you kind of the big picture, and then we'll dive into some of these little black boxes a little bit more to see how they actually work. One of these should be familiar to you, chemiosmosis. We already talked about chemiosmosis in cellular respiration. We already talked about electron transport in cellular respiration and how that is related to chemiosmosis. So if you've got that under control now, this part will be pretty straightforward for you. Light absorption. Have to talk a little bit about light absorption. And then the process of carbon fixation. This term, carbon fixation, basically refers to the fact that inorganic carbon in the form of CO2, which is in the air, okay, is converted by photosynthesis into carbohydrate, glucose. So basically we've got to add some hydrogen onto the carbon dioxide to make sugar. Don't bring the sand anymore.
I'm going to try not to.
Okay, if we look at cellular respiration, we've got two main processes that are going on. And we call those two main processes light-dependent reactions and light-independent reactions. In older textbooks, and Dr. Morton and I grew up with this terminology, they used to call break photosynthesis down into light reactions and dark reactions. That's a misnomer, because that really implies that this set of reactions only happens in the dark, in the absence of light. Actually, these are light-independent reactions, and I'll explain that particular terminology in just a second. Okay, but don't use this term, dark reactions. Okay, so the first three boxes right up here are light-dependent. When the light's on, this can happen. When the light turns off, this stops. This box down here, the carbon fixation box, which is also known as the Calvin cycle, does not directly depend upon light. It does, however, depend upon things that are produced in these light reactions. Okay, so think about that for a second. Light is necessary to make these things right here. This requires these substances. If we build up excess amounts of these during the daylight, then this can happen in the absence of the sunlight as long as there are these present. And that's actually what happens in photosynthesis. The Calvin cycle takes place in a region of the chloroplast called the stroma. And the stroma is kind of like the matrix of the mitochondria. It's kind of a fluid-filled environment inside the chloroplasts. The light-dependent reactions are associated with some membranes inside the chloroplast. And those membranes are called thylakoid membranes. And actually, these thylakoid membranes inside the chloroplast are stacked on top of one another, kind of like a little stack of coins. What is this stacking of the membranes an adaptation for?
Increasing surface area.
Increasing surface area. Okay? And why do we want to increase surface area? Because what's associated with the surface of these thylakoid membranes are pigment molecules, including chlorophyll. If we don't have chlorophyll, we don't absorb light. If we don't absorb light, this doesn't happen. If this doesn't happen, this doesn't happen. So the more chlorophyll molecules you can pack into the chloroplast, the more efficient the light absorption will be, the more efficient photosynthesis will be. Another thing that's associated with these thylakoid membranes is the electron transport system. And if electron transport doesn't happen, chemiosmosis doesn't happen, and therefore we don't produce these compounds. And therefore this stops. One of the other important reactants in photosynthesis that's involved in this light-dependent process are water molecules. And in the presence of sunlight, water molecules are actually split. Bonds are broken. And hydrogens are released, and oxygen is released. What happens to those hydrogens? They go through the electron transport system. As a result of the electron transport system, a hydrogen ion gradient is formed, and ATP is made by chemiosmosis. The other thing that happens is that as water is split, this molecule, NADP, becomes reduced. And then that molecule is then used in this carbon fixation process. So where do the hydrogens come from that ultimately end up in glucose? They come from water. Eventually they're added to CO2 from this coenzyme. And then CO2, when it accepts those hydrogens, is converted into glucose. What the light energy is really doing is two things. It's splitting these water molecules, generating the oxygen, generating the hydrogen that's used to reduce NADP, and it's kicking off an electron transport chain. And during electron transport, ATP is being made. So basically, light is causing, if you like, an electrical current to occur, and that electrical current drives ATP synthesis. Is that unusual that light can stimulate an electrical current? No, many of you have solar calculators, for example. You flip that thing open, and if there's light, there are metals in the light solar cells that absorb the light energy, and that causes electrical flow to occur. It powers your battery. In photosynthesis, when chlorophyll absorbs light energy, electron transport is initiated. And that electron transport causes ATP to be formed, and it causes NADP to be reduced. And we use those things in this carbon fixation process or the Calvin cycle.
So is the O2 and the hydrogen bonds just a waste product? Because it's coming off of the electron.
O2 would be a waste product. And in some respects, you could consider glucose to be a waste product or a product of this. Okay? So, here's a drawing of a cell, plant cell. See, there's numerous chloroplasts in this plant cell. There's our central vacuole, our nucleus, our Golgi apparatus, cell wall, okay? But if you look inside the chloroplast, you can see that the chloroplast has an outer membrane, and then it has these thylakoid membranes that are stacked together, and this is where the chlorophyll molecules are found. They're embedded into those membranes. And when light energy is absorbed by that pigment chlorophyll, several things happen. Water molecules become split. The oxygen, two oxygens from water, bond with one another to form an O2 molecule. As the oxygen builds up in the chloroplast, it will then diffuse out of the chloroplast into the cytoplasm. As it builds up in the cytoplasm, it will then diffuse out of the plant cell and be released into the environment. So the Earth's atmosphere, which was in ancient Earth history oxygen-free, accumulated molecule by molecule by molecule as a result of this process of photosynthesis. We're to the point now where the average concentration of oxygen in the atmosphere is about 20%. It takes a lot of photosynthesis to make that happen. During these light-dependent reactions that are associated with the thylakoid membranes and the electron transport system, we're going to make some ATP by chemiosmosis, and we're going to reduce a coenzyme. This coenzyme you can think of as kind of a cousin to NAD that we saw in cellular respiration. It has an additional phosphate group. But this becomes reduced during these light-dependent reactions. In the stroma, there are all of the enzymes associated with the Calvin cycle. And there's a bunch of them. We're going to talk about the Calvin cycle in a little bit of detail, but I'm going to really simplify it because biochemically this is a very complicated cyclical process. And all these enzymes are found in the stroma. So this ATP and this reduced NADP that's produced during the light-dependent reactions are now available to be used by those enzymes in this Calvin cycle process. What does the Calvin cycle do? It grabs CO2 and using this and this converts the CO2 into sugar. And in the process, like the citric acid cycle, it renews itself. As ATP is used, ADP is generated as a waste product. What's that used for? That's used back in the light-dependent reaction and becomes rephosphorylated back into ATP. The NADPH, when it's used, becomes oxidized. What happens to that? That's reduced again during the light reaction. So there's a real interdependence between these two sets of reactions. And we're constantly recycling our coenzymes, and we're recycling ATP to ADP back and forth again. What drives all that? Absorption of light energy by the chlorophyll. So how many times, given this representation of the Calvin cycle, does this cycle have to turn around to make a molecule of glucose? 6, right? Because the cycle's constantly got to regenerate itself. So if we're going to have a sugar coming out of the cycle, C6H12O6, we need to put in 6 CO2s. So we're feeding CO2 in. Where does it come from? The atmosphere. We're using water that the roots have absorbed from the soil and transported into all of the tissues, including the leaves. We're absorbing light energy with the chloroplasts and the chlorophyll. And that drives this process that produces carbohydrate. What do plants do with that carbohydrate? They can break it down catabolically to make ATP, cellular respiration. Or they can divert this sugar into other anabolic processes and make proteins out of it, make carbohydrates out of it, make lipids out of it. Sometimes they need some additional nutrients to make that happen. Remember that sugar doesn't have nitrogen. If you're going to make amino acids out of sugar, you need a source of nitrogen. That's why we have to fertilize plants to add some of those key nutrients to the soil. Not only is the chloroplast adapted structurally to the function of photosynthesis, leaves are as well. So, here's a typical twig with some leaves on it. Okay? And if we take a cross-section of one of those leaves, we'll see that it consists of several different layers. There's an upper layer and a lower layer to a leaf. And that's called the epidermis. That's not up there. So here's the upper epidermis up here, and here's the lower epidermis. Packed in between that upper and lower layer is the mesophyll. Phyll means leaf. Anybody know what meso means? Means middle. Okay? So these cells are part of the tissue that is in the middle of the leaf. And mesophyll cells are packed with chloroplasts. And then we have some veins in the leaf as well. Some tissue that transports water to the leaf. and can transport the sugar away from those cells that's produced during photosynthesis. Anybody remember or know the two types of tissue that are found in this vein?
Or xylem and phloem.
Xylem and phloem. Do you remember those? Xylem are the tubes that transport water, and phloem are the little tubes that transport food. It's kind of analogous to our arteries and veins in some respect. And then here is our representation of a chloroplast. And here you can see all those little thylakoid membranes stacked up. And what I like about this one is this shows you that the thylakoid membrane actually surrounds a little space inside of it. So we've got a space, a membrane, and then the stroma. So we've got a compartment, a membrane, and another compartment. What do we need two compartments separated by a membrane to do? Compartment, membrane, compartment. Hemiosmosis, right? And then here's an electron micrograph of that particular chloroplast showing you the stacked thylakoid membranes. Okay. Now, why do plant leaves tend to be broad and flat for the most part? What's the advantage of that? More surface area to absorb more sunlight. And are you going to absorb more sunlight and have more surface area if your leaves are all shriveled up or if they are erect? More if they're erect, right? If the cells are turgid, if they're swollen, and remember it's a cell wall that allows plant cells to accumulate a lot of water in their vacuole and swell, and that makes the leaf cells turgid and keeps the leaves erect to have maximum surface area. One of the things that leaves are notorious for doing is losing water to the atmosphere. On a hot, sunny day, especially when it's windy, a corn plant can lose gallons and gallons of water to the atmosphere. It sucks it up from the soil, and then it evaporates from the leaves. So leaves have evolved some adaptations associated with the epidermis. There's the upper epidermis, there's the lower, and then this is the mesophyll, where there's a little waxy coating on both surfaces of the epidermis called the cuticle. And that cuticle is a waxy layer that helps to retard water loss. So plants kind of waterproof their leaves. But there's a problem with that. If water can't get in and out, what else can't get in and out? Carbon dioxide can't get in. And we need carbon dioxide coming from the atmosphere into the leaf as a reactant of photosynthesis. And oxygen can't get out. That would be problematic for us because we need the oxygen to breathe. But also, as we'll see later, it's a problem for a plant if too much oxygen builds up in that leaf tissue. That can actually inhibit photosynthesis if too much oxygen builds up. So, there are these little structures inside, in the lower epidermis particularly, called stomata. And basically they consist of a couple little cells that can change shape depending upon whether they take on water or not. So swollen, not swollen. And that little opening can close and open depending upon environmental conditions. When would be a good time potentially to close these? when it's hot, when it's windy. How about at night? Okay, because the light-dependent reactions aren't happening at night, and the light-independent reactions don't happen very readily at night either. So closing those stomata down at night can be really an advantage. On Tuesday, we will talk about pigments. Talk about chlorophyll, the role it plays in photosynthesis, and also talk about some of the helper or accessory pigments that play a role in photosynthesis. This is a pretty good breaking point. Before you leave, a couple of things. On Monday, anything that you need to do in terms of lab preparation? If you want to take some time to do that, and then that will also be a time you could ask any questions, review questions for the lab quiz. Okay? There will not be a reading quiz on Monday. Okay? I'm not going to give you an exam on Friday and make you take a quiz on Monday. All right?
Just a reminder, I will be at the mentoring session Sunday evening at 7.
Oh, you'll be at the mentoring session Sunday evening in case there are questions about the lab quiz, right? Okay. And in lab next week, you'll take the lab quiz during your lab section, and you all should have gotten a study guide for the lab quiz, right? Okay, I emailed that to you. I think we'll do that first probably, right? And then after you take the lab quiz, you've got the remaining of the lab period to work on your data and to start working on your presentation, because you have to do your presentation when you come back from break. So we'll do a combination of review and then a little preparation for next week's lab in Monday session. Okay, so we'll pick back up here on Tuesday. Okay? Thank you.
I have a science exam.
Oh, thanks.